Hello everyone, and a very warm welcome to this particular webinar session on the AutoDock Vina 1.2, the new version. I hope I'm audible to all of you. Kindly confirm, one of you can confirm the same in the chat box so I know that you are able to hear me clearly and also able to see the screen, the title screen of molecular docking with new Vina 1.2. We will start the session in another 2 minutes only and we'll start discussing about Veena. Hello Najiba, welcome back. All right, that's really great. And I'm really happy that you all have joined over here. I really welcome all of you back over here. All right. Okay, thank you so much for the confirmation. Hello, hello. Hi everyone. It's really good to see you all of you. All right, I think we can get started with this particular session and let's get started. So, hello all. My name is Jitesh Doshi, and I'll be your instructor for this particular session, which is on AutoDock Vina 1.2. As it has already been advertised, this particular session is about the new updates, the new features which have been added to the recent version of AutoDock Vina, which is one of the most popular molecular docking tool out there. So let's get started slowly. Before we go into the details of all these things, let me clarify a few things. It is expected that you do have some little bit idea about the Autodock Vina and some basic command line usage. But even if you don't worry about it. I will be able to answer your questions and will go in such a way that you will at least get some clarity. Second thing that I'm sure that you're gonna have lots of different types of questions regarding the molecular docking, not just about the Vina, but in general the preparation and all. So my one sincere request to all of you is that uh, if you have questions directly pertaining to whatever I'm saying, talking, or showing you on screen, then go ahead and ask me in the chat box right away. All the questions which are not directly related or they may be somewhat peripheral, I will keep it at the end. We have a Q&A session, so either you can ask them at the end of the session during the Q&A, or even if you post them, I may miss them, so better to keep it and ask them at the end of it. So we will discuss those as well. In this, the agenda for this particular course is going to be introduction to what Autodock Vina typically is, what has changed from what has been there for last 10 years, and this year what new things have been introduced, how to install different components which are required, and how to set up and get your PC ready for using all these different tools. I will also demonstrate utility of some of these tools which often are required by many people. And if time permits, we are within the time limits, then I will discuss a slide about how to set up a Python environment. I am assuming that not many of you will be familiar with the Python at this point. So I will keep it as an extra thing to be discussed at the end. And then followed by that, we will have a detailed Q&A session where you can ask me questions about this particular software as well as other things that you might have in your mind about the molecular docking. I will not be focusing a lot on the preparatory steps, how to prepare the molecules, what care to be taken, because that's not the point of discussion today. I solely will focus mostly on the features, technical features of the Autodock Vina, along with some demonstrations of the molecule. So obviously in this process, we might be discussing some parts of the preparation and so on. So let's talk about that. So let's get started. AutoDock Vina is one of the most popular algorithm for performing molecular docking calculations and that has been there for a really long time. The earlier version of Vina, that is Vina 1.1.2, was last introduced into the 2011, and after that there has been no updates directly in 2021. After 10 years, we have the new updates to AutoDock Vina, which combines lot of good and utilizable features into one packet. And that's the reason why I have planned this workshop, so all of you know about what is the new AutoDock Vina 1.2 is all about and what can we do with that. So let's get started. AutoDock Vina is a very fast algorithm. It uses Monte Carlo simulations to optimize the poses that we need. So for those who might not be really familiar with the autodock or docking concept in general, just one line introduction to it. Molecular docking is a study of interactions between the two molecules, maybe two biomolecules like proteins, maybe one small molecule and one macromolecule like protein and ligand or protein and drug. And by using the molecular modeling techniques, the interaction between these are introduced or calculated by using docking methodologies. And to perform these calculations, we have AutoDock Vina as one of the tools. Many of you may already be familiar with the older version of Vina, which has been popular for a decade. And it looks something like this, which you can see. It is a command line tool, or most of the Autodock related tools will be command line based. So this is how it used to look. It has this interface. The new version is also very similar. So first, we will talk about the differences between these two versions, what things have changed and what new has been added. So if you look at the history of Autodock entirely, well, it has been changed quite a lot of time. It has started with the one version which was introduced quite early into the docking developments, and the most stable and the popular version has been the 1.1.2. As I mentioned, it has been there for last 10 years. But this year, the authors of Autodock Vina, they introduced a completely new and refactored version called Autodock Vina 1.2.0 and the sub versions of those. So that's what we will be focusing on today. So what is the new in this update? What additional things have been taken into account? The first thing is the choice of scoring function. So in docking protocols, there are multiple different types of scoring functions which use different methods to score a particular pose in order to get a docking score or affinity or binding energy. So in earlier Autodock Vina, a default Vina function was used, but now you have a choice of using three different functions. One is Autodock 4, which is another popular scoring function from Autodock. One is the default Vina function, and the third is the Vinardo, which is slightly modified, improved function for scoring the poses. So you can now choose any one of these scoring function to score your poses and then analyze your data. This also has an ability to read and write external maps. What it means is, in earlier versions of Autodock Vina, the affinity maps for every atom type in a ligand used to be automatically created and used during the docking, as opposed to the Autodock where we had to create these maps manually. So now in this version, all the affinity maps generated by the AutoDock algorithm can be imported into AutoDock Vina, and they can be used with the AutoDock function. These internal maps can also be written for use in some other tools. The most important and very useful addition has been the batch mode. Many people over the years have faced this problem of using the AutoDock Vina command line for running the virtual screening experiment, where you want to dock a large number of compounds or a library of compounds in one single go. Fortunately, in this upgrade, you have the batch mode where you can perform this easily just by single command by introducing all the ligands that you want to dock against your protein. So this batch mode is going to be very useful for many of you who want to screen the large number of compounds. Another additional feature that the AutoDock Vina update has is a simultaneous docking for fragment-based drug discovery. So simultaneous docking means that at the same time two different ligands can be docked into the same binding cavity. This has good applications in the fragment-based drug discovery. Again, for those who are not aware of what fragment-based drug discovery is, what typically is done in this case is, if this is your binding site, you can at a time give two different ligands to be docked, and different positions of both the ligands simultaneously will be explored in order for you to give the best poses for each one of these ligand, which then can be used to join together and create a novel inhibitor or a novel ligand for that binding cavity. So this fragment based docking can also be done now by using this new AutoDock Vina 1.2 upgrade. Earlier, to perform the docking with the zinc metalloproteins or the proteins containing zinc or metal ions, you had to use a specialized AutoDock zinc force field, that too only with AutoDock. The AutoDock Vina earlier was not capable of doing this, but now they have introduced this facility within the AutoDock Vina itself, so that you can also perform the docking of your ligands with zinc metalloproteins. We'll discuss a little bit more about how it is done and what can be discussed in this regard. Apart from this, a very useful tool, that is the hydrated or a solvated docking, can also be performed. So one of the limitation of the general docking protocols is that we do not consider the solvent, we do not consider the water molecules to play a role in the binding. That's why the hydrated docking, what it does, it also creates a kind of sphere of water molecules around your ligand. It hydrates it, and the hydrated ligand is then docked against the protein to find out if any one of these water molecules are really important for the more tighter binding of this ligand or not. So this hydrated docking can also now be performed with this new AutoDock 1.2. Along with this, the most useful feature is the Python integration. This AutoDock Vina can directly be imported as a library into your Python code. So if you are a developer or if you want to automate the process of molecular docking and analysis, and if you're using Python as your environment, it's a programming language which can be used for multiple different scientific computing tasks. So this can be easily integrated into this Python. It also comes with a Python-based advanced ligand preparation tool where we can titrate the molecules at a certain pH, make sure that the ligands are prepared, and this also will be important for some other things like hydrated docking, and it also has an additional support for macrocyclic compounds. This is something which was missing earlier. There are a class of compounds which we call as macrocyclic compounds. These contain a very large, maybe 16 member, 18 member, 24 member rings of carbons, and these are highly flexible. Earlier protocols limit the rotational degrees of freedom to around 32 rotatable bonds, but that cannot be applied to this because these are highly flexible. So they have included a very optimized scoring function to be able to get the best poses from these highly flexible macrocyclic compounds called as macrocycle docking. So both of this can be performed with the Miko. So these are the new things that you have. Now, what changes are there into the interface or overall look of the tool? Well, there are not much. Both of them still are the completely command line based tools. So I will very quickly go over some of the differences in the additional features that you have. So let's say what differences in the interface has been there in the input as well as the search area. So in the older version of Autodock, that is Vina 1.1.2, we had these options for input like receptor, the flexible receptor and the ligand, and the search space could be provided by the center and size of the binding box or the search box. In the Vina, new Vina, we have a lot of other options as well. In the input, we have obviously the three minimum options are there where we can provide the receptor, the flexible part of the receptor for flexible docking and ligand. But along with that, we have two more inputs that we can provide. One is the batch mode where you can dock multiple compounds at the same time, and the scoring option where you can choose which scoring function do you want to use, whether AutoDock 4 scoring function, Vina, or Vinardo. The search space obviously has the same coordinate system to be used over here, but now you also has option to include the external maps like AutoDock 4 maps, affinity maps, which you can be prepared to use for AutoDock 4 function. And you also have a simple way of automatically generating this particular grid box by using AutoBox. So if you already have one ligand which has been either manually fixed into the binding cavity or it is a co-crystallized ligand, you can directly use that ligand to create a box by using the AutoBox. So this automatically will be generated by using the new Vina. So these are some of the improvements and problems where people have faced have been answered with this. The difference in the output are not much. You still have the same optional data to be put, like the name of the output file, the name of the log file, and so on. In new version, you still have the output option, but along with that, you have two new options, which is called as the dir, which is a directory. So if you want to perform the batch mode, you can create a new directory where all the results will be saved automatically. And write maps, so if you want to write the Vina maps to an external file, you can always use this option. Okay, there is a question. Is there new option to change the spacing since the spacing in the old version is fixed in 1.1 angstrom? Yes, absolutely. So in this new version, the default spacing is just like AutoDock, that is 0.375. And by using the hyphen spacing option, you can change that to anything that you want. Yes, you do have that option. That's a good question. I'll show you now here is in the miscellaneous that. Then look at the miscellaneous options. So earlier we had some very basic option as changing the number of CPUs that you could use, the random seed that you can put to start your randomization, exhaustiveness of the algorithm which sets how many times the global search should be kind of performed. The Monte Carlo simulation should be performed so that your chance of getting the global minima will increase. So the default values still stay the same, that is 8, and these options are the same. You also have the energy range to be put and the configuration. But now we have some other options as well to be considered. We have the maximum evaluations that we can do in every run. We can change that. So we can set up our own customized docking protocols in case you require. If you're not familiar with these, you're okay to just keep it default values and not give these things. Absolutely fine. As just asked by Najiba, the spacing option has been given. The default is set to 0.375. Earlier, it used to be default 1 angstrom, and you always required that, but now it can be done at the higher resolution for better results. And the configuration file can be done or given in the exactly same way, where all of these options can be written in a single file and given to the input command rather than giving all of them individually on the command line. And finally, the most important changes have happened to the advanced options. The advanced options in the earlier version of Vina were specifically the scoring function that can be tweaked by applying different weights to different types of interactions like repulsive interactions, hydrophobic interactions, hydrogen bonding, rotational degrees of freedom, and so on. But now, as we have three different scoring functions that can be included, you have these weights supplied for each one of the scoring function individually that you can change. This is only in advanced cases when you know what you're doing and you understand the repercussions or the complications that could be caused with these kind of changes. So, but these are options available. Apart from that, you also have options to perform the only scoring of the already docked poses or crystal poses, as well as only perform the local optimization of the molecules as well, and some more function to perform the grid generation properly. So these are the updates or the changes that has happened to the AutoDock Vina from the earlier popular version to the new version, that is 1.2.3, which is currently the latest one. All right, now let's get to the little bit of technical stuff To be able to get the full utility of this entire AutoDock Vina ecosystem, only AutoDock Vina I'm talking about here, not rest of the AutoDock ecosystem. So we need to get ready our computer with multiple different components for different purposes. Here I have listed some of these components. I will also tell you how to install them and where to install them from, as well as what can be skipped in certain cases. Because uh, if you are very beginner to the docking, this could all be very overwhelming, very complicated it might look. So I want to make it simplified for most of you. That's why I will also mention the alternative options. Now, one thing you always need, no matter which program you're using, you need some kind of a molecular visualizer and editing tool like UCSF Chimera or Discovery Studio or PyMol or Autodoc tools or MGL tools from which you can do your basic preparation, like deleting up some of the chains or identifying the crystal poses or removing some unwanted molecules or ligands and so on. So for this basic preparation you are going to need this. Again, to add the hydrogens also you're going to need these tools. So if you have any one of these tools already with you, I think you're sorted for this part. The second thing or second software that you need to install is called as ADFR. It stands for AutoDock Flexible Receptor. So this is a better version of AutoDock in which you have some very useful scripts, useful tools. For AutoDock Vina, which of these will be useful? Well, it contains AutoGrid 4. So in case you want to include the affinity maps of AutoDock or AutoDock scoring function, you will need this AutoGrid option. So this is only when you want to use the AD4 scoring function. This will not be required if you are just going to go with the normal standard Vina function. It has two other very useful tools, which is called as prepare receptor script and the prepare ligand script. Both of these are executable Python scripts which can help you prepare your molecules right from the command line without having to go to any other graphical tool. So this is a lot of general options that you might need, like adding of hydrogens, removing of non-standard, merging of non-polar hydrogens, and so on. Basic preparation that we need to do can be done with this. And along with that, ADFR also has many additional useful tools like the Autodock crank pep, which is for peptide docking, and many others. I'll show you during the visualization of these tools. The next components you will need is the AutoDock Vina 1.2 scripts. For some specialized docking protocols like hydrated protocol or zinc metalloprotein protocols, AutoDock Vina 1.2 comes with some pre-written scripts to make your life easy, which can be downloaded from their GitHub page. I will show you that as well. But for simple general docking, you may not need all of these. OpenBabel is a very well-known molecular file format converter which can be used in order to perform the batch molecular conversion. So if you have like 100 or 1000 molecules, you want to convert or optimize them all at the same time and convert them into the PDBQT format required by Vina, you can use OpenBabel command line options. Also, the OpenBabel is required for the Miko package, which we will talk about, that has been introduced along with this new AutoDock 1.2. If you want to implement a Python support or a Python language in implementation of Vina, you will also need to install that Python language. I would recommend you to install it through an Anaconda, which is one of the Python interpreter. It has all the necessary things all set up and it's easy and compatible with most of the libraries and other tools. So you can use this Anaconda installation to get the Python language. This will be required for Python integration of Vina and as well as for the Miko tool that you need to install. This Miko Python package then can be also installed individually. And finally, the most important part, so this Miko tool, it will be required if you want to perform the macrocycle docking as well as hydrated docking. If you don't want to go for these specialized protocols, you don't need it. And finally, we need the actual AutoDock Vina 1.2 executable file or the main program that will perform all of these calculations. Now, out of this, which is mandatory and which is optional? Well, if you are going to only perform the most basic type of docking with some few molecules or maybe several molecules, you can skip installing the Python as well as this Miko package, which will not be important if you're not going to do anything with the Python programming or this other type of docking. So this can be skipped. If you are still going to go for a very basic protocols, even these scripts can be skipped from downloading. So only thing you will need is the some program to visualize your proteins, ADFR to prepare these things. If you already know how to use the AutoDock tools to prepare your receptor and ligand, even this you can skip. So only thing you will need is some program and you will need the AutoDock Vina executable. So that's the only thing you're going to need for installation. But if you want to perform the full capacity or explore the complete utility of this tool, all of this could be installed and get ready. Now, I'm gonna very quickly go over these things one by one and show you where to get them and what should be considered while installing them one by one. So this is a preferred sequence, but obviously it can change a little bit here and there. So I am going to use for visualization purpose a tool called UCSF Chimera, which we can use for basic preparation and identifying the binding site or the coordinates of the binding site and so on. The This is the website for I will be sharing this presentation in the description at the end of this session, so you can get this link directly. So here is the UCF Chimera link. According to your system, you can download the version. It's freely accessible and get it installed. Then ADFR suite can also be directly installed from the Autodock website or the Scripps Research Institute website. This ADFR contains many useful tools that you will need during your docking calculations. So you can download this directly from this particular website, and during download there is nothing much you have to consider. It's a direct executable. Simply go to the particular system. I'm assuming that most of you are going to use Windows, so that's why I'm going to show most of the things on Windows, but it is it will be really good if you can also explore the Linux options. All of these things work much better way in Linux. So you can download the ADFR suite from the link given, and you will get lots of different things along with this. So installation will be in your program files, and it has lot of things you might need, like AutoGrid or AutoDock, and then ADCP, that is the Autodock crank for peptide docking, and these important scripts called as prepare ligand and prepare receptor. They will help you prepare your molecules for the docking. That's about the ADFR. So again, easy. OpenBabel is also very easy and straightforward installation. You simply have to go to the website, download based on your system appropriate version, and just simply install it just like any other software. Now comes the time for Anaconda. Again, if you're not familiar or not planning to use the Python at all, then you can skip this option. But if you want to perform all the advanced docking as well, Anaconda installation would be helpful. Again, installation of Anaconda is very easy, but using that could be difficult for beginners. I will maybe take another session specifically for these setup of Python environment for those who are interested. And finally, let's come to the main program, that is our AutoDock Vina 1.2, where to go, get it from and what to look for. So the AutoDock 1.2 is available as an executable on their website or the GitHub page of the AutoDock Vina. The latest version is 1.2.3, and here you have three executables for Linux, Mac as well as Windows, all 64-bit versions. So according to the system that you're using, you will be able to download these things and use it. It is a simple executable file directly, so you don't need to install it. Just keep it in a safe folder somewhere from where you can easily access it, or you can add it to the environment variables for quick access. So this is where you will need to download it. And the scripts that I talked about the AutoDock Vina, you can go to this main page AutoDock Vina, the home page of the GitHub directory, and simply download this entire content. So you can go to the code and download zip. So this zip file that you will download will contain all of these different things like this, which has the builds for different versions. It has some useful data like parameters or the force field parameters. It has some example files that you can explore, and it has some pre-made ready-made scripts. So these scripts will be only required if you want to use the AutoDock scoring function, or you want to prepare the flexible receptor from command line, or you want to do the water mapping, that is the hydrated docking, or you want to perform the zinc atom or the zinc force field related calculations. So I will be showing you all of this. So this folder will also be very handy for you to access this ready-made script and make it easy. So technically most of the stuff we'll be discussing here are going to be on a command line. So I hope most of you are familiar with the command line utility of the AutoDock Vina. So that's about the things that you're going to need. And finally, once you install this Anaconda, you can install the Vina Python version as well as the Miko tool for preparation of certain molecules through Anaconda. So that's overall going to be installation looking like. So it's not that difficult. It's just that there are lots of components you might need for different purposes. Again, I'm emphasizing that for the most basic simple protein ligand docking, a simple executable is more than enough. Just prepare normally the way you prepare your molecules and directly give it to the executable Vina, and that's all you can do. But if you want to do any other type of docking, you will need all of these different options. Okay, so I hope this part is clear, like what are the requirements, what is the environment like, what new things that you can do with this. If you have any questions or queries, let me know. Now we will take from the simple to little bit advanced, one by one, different types of docking protocols, and we will perform them quickly. The preparation I will do in front of you, but I won't be discussing much of the preparatory steps because that's not what we are meeting here for. Okay, let me know if you have any questions. Meanwhile, I will open the things. Okay, so let's start with a very basic, simple docking protocol. This includes preparation of the protein with any visualization tool. This basic preparation would include things like removing the unwanted things, adding the hydrogen atoms as per the protonation states that you desire, or a pH at which you want to titrate your protein. So this sanitization will be very useful for clean and good docking results. The AutoDock Vina expects your protein files to be in a PDBQT format. So this PDBQT format can be converted from PDB by using either AutoDock tools or the ADFR prepare script, so on. You can use any one of these tools. Okay, there is a question from Roshan. Is it always necessary to check for missing atoms of a protein during protein preparation before docking? What is the best way to fix the protein is having missing atoms? Yes, it is absolutely essential to treat the missing atoms, because even a single atom that is missing, if it is contributing towards the interaction, that's going to completely ruin your results. So it's very important to check for missing atoms of all the amino acids. One of the simple ways to do it by using this tool called as AutoDock Tools. AutoDock Tools has an option to fix the missing atoms from your amino acids, so it can do that easily. So you don't have to really worry much about those. Right. Once you convert your protein into the PDBQT file, that is one input that we require. The next we need the ligand atom, the ligand molecule. So we need to prepare the ligand. Again, we can use either the AutoDock tools graphical interface if you are comfortable with that, or you can use OpenBabel if you have lots of different molecules to be converted at the same time, because one by one doing them in ADT might take some time. So you can do that in a batch mode in OpenBabel, or you can use the ADFR or MICO scripts, which are specifically for the ligand preparation. I will be using all the command line tools in this demonstration, but you are open to treat or generate your molecules from any one of the graphical interfaces. Then the next requirement for the Vina is the description of the search box. Where exactly you want to perform the docking, that is, where is your binding site. So this, we need to generate a configuration file which contains the size as well as the center coordinates of the search box, which can be generated either from AutoDock tools or from Chimera, or if you have a co-crystallized ligand coordinates, we can use those to auto-box our protein through AutoDock Vina 1.2. And then finally, a simple general Vina command, the way we have been used to, can be used to run the docking program. So let's start looking at it, how to set up and how to look at these software and so on. So let's close this. Okay. So the first thing I will do is on my desktop, I will create a new folder. Let's call it as demo. So I will save all the files over here. So for the basic docking, the first thing we're going to need is the uh, executable file. So what I will do is I will directly download it from here, as I'm currently on my Windows system. So I will use the Windows version of executable, save into the folder that we just created, that is a demo folder, and click on save. So now if you check the folder, you have the executable file, which is a Windows executable file over here. To access this, we can directly use it from the command line. I will show you how that does look. We are going to need a protein and a ligand. Okay, another question. Can we use AutoDock tools to prepare PDBQT files for both receptor and ligand? Absolutely, yes. So you can use the AutoDock tools as well to prepare your proteins and ligands files easily. That's graphical interface, so that would be easier. Okay, I can do that. I can use that as well to prepare these files while showing you. All right. So now we have the Vina. Now we need a protein and a ligand, and I'm going to use Chimera to essentially prepare the basic things about my protein and use it. So the protein that we're going to use as an example here, let's download from the PDB. It's going to be a kinase enzyme, the PDB ID of which is 5LXC. In case you are also performing these with me, try to do it, but as we are going a little bit faster, you may lose the track of what's happening. So I would recommend you go through this video one more time and try it on your own. And obviously any questions you can post on the comment section of this later also after the video ends. It will be anyways available on the same link after this as well. So let's perform. Can you please use multiple ligands for docking now? No, I'm going to go one by one, but I'm going to show you all. A single ligand, multiple ligands, simultaneous docking. I'm going to show you all different types, but one by one. So because there are some beginners as well, so we can't skip any of these steps. Okay, so the system we're going to use is this protein called as 5LXC, which is a DYRK2 kinase enzyme, and we will perform a simple redocking experiment by separating the protein and ligand, and then we will perform the docking of that again to find their affinity. So I will download the PDB file and save it in our demo folder, which is the 5lxc.pdb. To check the file and do some basic preparation, I will go to the file, open demo folder and open the 5lxc.pdb. Now looking at the file, the protein contains two chains. We can only work with the single chain for now, so we can delete one of the chains. So for that, I will go to select chain and select one of the chains. If you're familiar with any other visualization tool like Discovery Studio or PyMol, you can use that as well. I'll go to the actions and delete that chain, so I can only work with this. Now this chain contains a lot of other things like many other ligands, one co-crystallized ligand, there are lots of water molecules, and there are no hydrogens. So we need to do some basic preparation as well here. To perform that, I will go to select residues, and I will select the residues like water molecules. Again, delete them. There are some simple options as well, but just to show you step by step, I am going one by one. Then we have these ethanediol molecules in the protein. I'm going to delete them. Now we are left with the protein and the ligand which is there in the binding site. This is a ligand. This information obviously you will get from the PDB page, that what is actually the ligand of active investigation or LOI and so on. So you can get that information and map it over here. So now here we understand where the binding site is, where the ligand is bound. So we need to separate these two molecules from each other so that they can be saved separately as protein and ligand different. Why do we delete? Okay, there is a very good question. Why do we delete chain B, or why do we keep single chain for docking? Well, it depends a lot on your protein, individual protein. In this case, I know that this protein is made up of two identical chains, that is chain A and B, which are both identical, and each one of the chain has a binding site for the ligand. That means they are identical, and docking we can only do with one single chain at a time. In case your active site is present at the interface of the both the chains, then we need to keep both the chains. So you need to check that quickly before you process. Yeah, I think the next also is answered. If the target protein has iron molecule which it uses for its activity binding with its substrate, should we keep that molecules while docking? Yes, that will be the part of our metal ion docking protocol. I will come to that in some time. Right now, I'm just discussing the most basic docking. So I'll come to that question later. Okay. So now that we have it, we need to first of all split these two molecules from away from each other while keeping the ligand coordinate same. So what I will do is I'll go to command line over here and simply give a command called as split ligand, or you can manually also split them, separate them together by any other software and enter. So what this will do is this will create two separate files, one for the protein and one for the ligand. So first let me save individually each one of these things. So first let's select the protein part, go to file, save PDB. I will call this simply as protein, and here you can see protein and ligand, both of those are there. You can only select the protein part to save and simply click on save. Then you can again go back to the save PDB. This time you can only select the ligand part, the second part, and save it as ligand.pdb. So both these will be now saved. If you check the files, the new files that has been generated, we have a protein file, PDB file, which only contains the protein coordinates of different amino acids. Again, as I mentioned, I am not discussing the preparatory steps, so I'm not going to go into details of any one of the other things like sanitation and sanitization and all that. So, and this is the ligand molecule which only contains the heteroatoms. That means we have successfully separated the protein and ligand, but even if they are different already, it's a good thing. The next thing is that we need to convert them into PDBQT to be able to useful for docking. So that you can do by using either the scripts which come with the ADFR that you have installed, or you can do that by using AutoDock tools if you're comfortable. I'm going to show with the ADFR tools how can you convert this into the PDBQT files. The next thing we are going to need is the coordinates of the search box that we want to keep. So how do we generate the search box? Okay, another question. What if conformation of these chains are different? Can that be the case? Absolutely. If your protein is a heterodimer or heteromultimer, each chain is going to be different. So you need to first of all find out where exactly your active site or a binding site is present. If you're not sure, you need to perform the docking on each one of them. Again, these questions are, we will discuss in a separate session. I will tell you all about it. Can I use Linux OS for doing same? Absolutely. That's in fact much better and preferred way of performing these things. It will be easier, but it will be the same way. There are not going to be any differences as such in the commands. Okay, so now we have the protein PDB file and we have the ligand PDB file. Now we need the description of the binding site. To generate this box, I'm going to use this grid box by using the AutoDock Vina plugin of Chimera, where you can simply draw the box around the protein. The box that you often need, it needs to be around the binding site which is known. If not, then you have to understand the important residues of active site and use this. So this should be big enough to fit lot of different sizes of molecules. If it is too small, many large molecules may not fit for docking. If it is too big, it might take a long time or might give you the wrong superficial results about the docking. The recommended size maximum is 30 by 30 by 30. My current box dimensions are this. The center coordinates are 46, 12 and 16, while the size is around 21, 17, 19. Size we can vary a little bit. So now let's perform all the operations we need. Let's first create the binding configuration file that will be needed by the Vina. Again, I am hoping that you might have already used these kind of configuration files earlier, but if not, it is nothing but a very simple text file so that we can store all the information. If you don't want to create it, you can directly give the coordinates as well as size on the command line. But to save that, I'm saving this data. So it only needs you to give parameters like size in the x direction. I'm going to keep 22 roughly in each direction, in y direction also, and size in z direction or z coordinate also 22. But center cannot be changed. It has to be properly placed around the ligand. So center coordinates of the x are 46.29. Center y, as you can, I'm looking at this particular box and writing these values. Center of y is 12 and center z coordinate is around 6. This is with respect to the protein, so it needs to be prepared. This we only need to do once so that we can save this data. I'm going to save this file into the same folder and I'm going to call it as configuration. This will be saved as a text file or .txt file if you're using Notepad. That's it. So once we set up our proteins binding site by using this, we are all set to perform the docking. Now we need to convert the receptor and ligand into PDBQT files. So now let's access the command line and talk about all the things we need to do. Now onwards, it will be much more easier. I'm going to use the Windows command prompt, but you can use the Linux bash shell as well for the same. Before I start with the preparation and the running, let me quickly answer some questions. How can we find the active site of a novel protein? There are many algorithms available. Ones I would prefer are the AutoSite or AutoLigand, which are developed by the AutoDock community. So you can use those to predict the potential active sites in a novel protein if it is not known. In case of multiple chains in protein, which chain will be take for docking? That's completely up to you what you are looking for. So for example, one chain is doing a certain function, another chain is doing a certain function, and depending on which function you want to inhibit or which function you want to modulate, you will need to choose your chain. That cannot be generalized in any case. Can molecular docking process be performed with high molecular weight polymer like hydroxypropyl cellulose? Yes, you can do that. It does require some amount of modifications of these protocols. Specifically, you need to increase the exhaustiveness of your algorithm in a much higher capacity. It does take a lot of time, but you can easily perform those. Absolutely. There is no such thing that you cannot do. It's just that they being very flexible, you need to allow them to explore lots of different conformations. Is it acceptable to perform blind docking if we do not know about the active site? Yes, that's a general practice. If you're not aware of the active site or a binding site, you can perform the blind docking. That is, the complete protein will be inside the box. How can I find binding site of a protein? Let's discuss it separately. Can you make a separate video tutorial on covalent docking, induced fit docking, and autoligand? I'll try to definitely do that. But again, today I just wanted to discuss this new AutoDock tool. So let's focus on that quickly and come back to these questions at the end. But covalent docking can be done with AutoDock. Induced fit can also be done with AutoDock. Okay, so now I have opened my command prompt from where I can access these different things. The first thing I'm going to do is access the folder where my data is saved. So for that, I'm going to use the cd command, that is the change directory. My folder is on a desktop and the name of the folder is demo. So I will enter. This will make sure that I have access to all the files currently present inside this particular folder. If I use the dir command, it shows me list of all the files. So we have the Vina executable, we have the protein PDB, we have the ligand PDB, and we have the configuration.txt. That is the configuration file. So everything we need is ready. Now let's start preparation. First, preparation of the receptor. You can do that. That is the conversion of the protein into a PDBQT file. So we can do that either by using AutoDock tools, or we can do that by simply using a command called as prepare receptor. It's very easy to do. This command is automatically installed when you install the AutoDock FR or ADFR tool. So as I showed you earlier, when you install this ADFR software, it automatically installs these scripts called as prepare ligand and prepare receptor. If you directly run this program without giving any input, it will show you all the options that it can take. As you can see, it can uh, prepare your molecules, it can add hydrogens, it can rebuild the bonds, it can check for hydrogens, and so on. It also has options like merging of hydrogens, or if there are any lone pairs, those can also be merged. If there are any non-standard residues might be left, you can remove those by using this. So it's very simple, but we will use it directly by the default option. We already have added the hydrogens. Oh, sorry, we have not added the hydrogens. So we will need to do that. So for that, we're going to use this option, and that's it. So let's run this command. I will increase slightly the font size so you can see what's happening. So first is preparing the receptor. The receptor file name is our protein.pdb. We want to add hydrogens to this, so we will use this option called as -a hydrogens. This can be found out over here. So we want to add hydrogens, that's why we have used this, and that's it. Then we need to give the output file name. Let's call it as protein h.pdbqt. that means hydrogen added protein, or simply protein.pdbqt is fine. Any name you want to give, you can give, and that's it. The moment you run this command, it will create a file which is called as protein.pdbqt. As you can see, it is now in the list. So we have successfully converted our protein PDB into protein PDBQT. Exactly the same way, we can use the prepare ligand command to convert the ligand into PDBQT. So we don't have to really open any other software. You can do that all from the command line. Prepare ligand, and you can again see the options. It has lots of things, like it only requires the ligand file as an input minimum. Apart from that, you have adding of hydrogens if you want to add. So we will again add hydrogens. You have other options like rotatable bonds, assignments, and so on. But again, we are going to do the simple one. So prepare ligand. Our ligand file name is ligand.pdb. The only modification we want to do is add hydrogens. So it will automatically add the necessary hydrogens, and then the output file name should be ligand.pdb. pdbqt, and that's it. Run the command, and now you also have your ligand pdbqt ready. So now we have everything we need for Vina. We have a configuration of the binding box, we have the ligand pdbqt and protein. So we are all set to run the entire docking protocol. So we are going to use this new AutoDock Vina 1.2. The name seems very long, but you will have to use that, or you can rename the file to have a shorter name. What I'm going to do right now is to make it easy for you to understand. In the demo folder, this large file name that we have here, I'm going to simply change it to Vina 2, because I already have the normal version of Vina available. So I'm going to call it as Vina 2. You can call it some other things as well, what is good for you. And then we can use it. Okay. There are two questions. Can you tell me docking of metal complexes, transition metal with shift base with DNA or protein if time permits? Let's see. I'm not sure whether we will have enough time, but I will try to talk about it at least a little bit. May not be able to show it. Does it add only polar hydrogens? Yeah, no. Oh, by using the hydrogen option, it adds all the hydrogens. If you want to add only polar hydrogens, or what it does is it merges the non-polar later anyways. So you can use this option called as NPHs, that is non-polar hydrogens. They will be merged, but it will add all the hydrogens necessary. Okay, so now let's run the Vina 2 program that we have just downloaded. So Vina 2, if you, okay, sorry, we also have to give the extension .exe, so it becomes an Windows executable. Yep. And then we can run it. So when you run the Vina without giving any input, it will give you all the options that it can take. As we are gonna do the Vina with the minimum requirements, so we will build on the same thing with advanced things. So go one by one. So now we will see how to run the program. Simple. Vina 2 is our program name, so we are using it. It requires a receptor, so receptor file name can be given here. Our receptor file name is protein.pdbqt. Then it requires a ligand. Our ligand file name is ligand.pdbqt, and that's the minimum thing it requires. Same, just exactly same as the old Vina, and it requires an output file name that you can give. So let's call it as basic docking.pdbqt. So that's it. That's all we need to give as a minimum requirement, and then you can run. Okay, here we have a problem in one of the atom types. Which file? Okay, the hydrogens that has been added. So what we can do in this case is we can use the hydrogen addition, or we can skip the hydrogen addition and use it for direct automated hydrogen addition. So let me quickly re-prepare the receptor by using the option check hydrogens. So it will make sure that the hydrogens are correctly added. All right, prepare receptor. Okay, so it has now started running the AutoDock Vina just like a normal Vina program with the default setting low exhaustiveness, that is of 8, and it will give you the result and produce the output file just like earlier. So as you can see here, the energy that we are getting are positive. That means something has gone wrong, or either the binding site is away or ligand has not found the correct poses. We'll talk about that later, but this is how you can easily run that. Can we publish docking results performed through AutoDock Vina compiled in Chimera? Yes, you can do that. Anyway, you want you will have to cite both the tools. Some docking studies show that they perform energy minimization for ligands. Is it a good practice? Also, do we need to perform energy minimization for protein? Ligand optimization is absolutely necessary. It is a recommended practice for minimizing the ligand before docking. So that has to be done. But that is a part of the preparatory steps. So that's what I have not included that. But you can do that. If you're using a crystal structure, then you don't have to because it is already in an optimized pose. But all the other molecules you need to optimize. I'll show you how to do that. Also, do we need to perform energy minimization for a protein? If your protein is of a really good resolution, you may not need it. But if the resolution is not so good or some quality parameters are not good, or if you have modeled the protein, it's absolutely necessary to minimize the protein properly and then use it for docking. Okay. So now that we have this simple docking run, let's talk about some more options. I hope, okay. And then let's go for the multiple docking as well as the flexible docking. Which is the best for docking between AutoDock and PyRX? Well, PyRX is just a graphical interface. At the background of PyRX, it uses AutoDock Vina only. It doesn't use any new algorithm. It uses the same AutoDock or AutoDock Vina as we are using it individually on its own. It's just to make it easy for people to use a graphical interfaces for those who are not familiar with the command line. But again, there are some other issues involved with that. So I usually don't recommend using it right out of the box. So yeah. All right. So that's well, let's now do the advanced docking with multiple different molecules at the same time as well as the simultaneous docking. So first I'm gonna do is a quick rerun of this. Our box seems good enough. I think this should be able to occupy most of the common molecules that we might use. Let's quickly check our configuration file. All right. So I think this looks good. Now we can perform. So now that we have this files ready, let's go for the next docking. Now, again, the preparation of the protein is going to be the same. You can do that here. If you want to also add hydrogens right out of the box, you can actually do that here also. Here you have a better option of better control because as you know, hydrogen addition can be a very crucial part in how your docking results are generated. If you want to protonate your proteins or ligand at a certain pH or in a certain way, you have your manual way of specifying the protonation states for different amino acids like histidine, glutamate, and so on, and add the necessary hydrogens for all these molecules. The results were messed up because Vina 2 uses 0.375 grid spacing by default. Well, the spacing should not be a problem. The problem came because I did not add the hydrogens initially. That where the problem came from. So I will show you again by running the same thing by adding now the hydrogen. So this will be a better result. So I'm going to save first this as a protein PDB and I will also save the ligand PDB as ligand. Now that I have added the hydrogens, this will be able to run it properly. So let's quickly prepare our receptor without because we already have added hydrogens and then we can run it. All right. The same way we will also prepare the ligand. Prepare receptor, prepare ligand. And then we can rerun our Vina with the basic docking protocol the same way. So it's very important to add correct hydrogenation. Can you please add the command of spacing? Yes, I will do that as well. So as you can see, the default grid spacing is 0.375. Oh, okay. Still we have and to change that you can use simply a spacing option and you can give here So it will use the spacing of 1 angstrom instead of the default value, and that way you will be able to set up as per your requirements. I need to check why these are coming. The 0.375 should not be the reason for this because it's still a good resolution, so we can have that. So something wrong is happening. I think I have prepared or taken us some wrong files into this, but that's okay. We'll come about that there. Chimera dimensions are in 1 angstrom. Yes, that's true. That will be a too big a box, so that can be changed. Anyway, so now let's see how to perform the multiple or a batch mode, which is most desirable by many people when you want to perform the entire library of compounds and dock it. So for that, what I'm gonna do is I'm going to quickly get some molecules, maybe a few simple inhibitors we can take. So here I have a file of, I'm gonna keep the file into the same folder again. So the file I'm gonna use is this, that is the kinase inhibitors. As we are dealing with the kinase protein, I will open these kinase inhibitors and show you. There, I think there are 5 molecules, and if you want to prepare as well as uh, optimize as well as convert all of this molecule at the same time into PDBQT and run into a batch mode. Instead of 5, these can be even 500. The protocol and the commands will remain exactly same. They won't change. So I have here 5 molecules which we can compare. Now these are 5. So how do we prepare all of them? We did not give the configuration. Oh yes, I think that was very important. I forgot that part. Okay, thank you so much, Bharti ma'am. I'm gonna add that. Yes, because yeah, the configuration box was not given, so it was not able to decide where to dock the ligands. So we will just do the same thing with the configuration box, and that's will take care of your entire setup. So you have here your affinities of the molecule, and that will run. Now that we have for a single molecule, let's see how to perform for multiple molecules at the same time. So we have here a file which is called as kinase inhibitors.sdf. Now this is currently an SDF file. So we need to convert it into PDBQT, and also as a requirement, we need to optimize these molecules properly. That we can do by using the OpenBabel. OpenBabel, as I mentioned, is another convergence software which can also optimize the molecules. So to perform that, we will go ahead and use the OpenBabel command. It is automatically installed when you install the software. So this will minimize and convert all the molecules at the same time into multiple PDBQT files. So OBabel, then we have to give the options or input PDB file. Our input file is SDF file. So I will give the name of the file. Then we can also give the output PDB file. We need a PDBQT. Then we can give some options like if you want to minimize, if you want to give a specific force field for minimization, you can also give like mmff94 and so on. And then we have to give the output file name. Let's call it ligand.pdbqt. But if we run this basic command, it will only generate a single file, PDBQT, containing all the ligands. But we want multiple files individually for each ligand. So we will simply add this flag called as M. So for each one of the molecule from the list, it will generate individual file. And then you enter. Once you enter the command, it will first optimize the molecules, and after optimization, it will convert them into PDBQT. Optimization might take some time depending on, you can see here that the 5 molecules have been converted. It cannot use MMFF94, so it will use the default force field that it has. So if you check now the folder, ligand PDBQT, what name did I give? Ligand, yeah, ligand.pdbqt, that Oh, okay. I replaced actually the earlier file. It was the same name. I'm just gonna call it again a simple molecule, and I'm gonna use a flag M for multiple. It's a very simple command that you can learn separately as well in order to perform this. So here we have 5 molecules converted and five output files will be generated. So for each one of the file, you have this name molecule 1, 2, 3, 4, 5. Now to use the batch mode, this will be very helpful having some kind of a pattern in a file name. If you don't have a pattern, every file has a different name, you will have to give every name one by one. So it's better to have a pattern so you can give all the files at the same time. Once you do that, the next step, again, I will give you these steps as well that I have written over here later at the end. So we need to create a directory to store these results. Let's simply call that directory as results directory. So all the results will be saved into this particular folder. So once you do that, now your command will be like this. We have Vina, then we have receptor will be the same, that won't change. Let's call it as protein.pdbqt. Instead of ligand, what you will use is, instead of ligand, you will use a batch option and you will use the pattern that your molecule files has. Our names are molecule 1, 2, 3, 4, 5. So this 1, 2, 3, 4, 5 are the only differences which are covered by this asterisk, which is the wild card. So it can take any value. So it will look for all the files matching this description in a batch mode. Then we have to give a directory in which this results needs to be saved. We have created a directory called results, and finally we'll need a config file to get the configuration. So this is a very simple batch mode where once you give this, simply enter and one by one it will take all the all the molecules to be used in this molecule. Okay, I think spelling is correct. Yeah. Okay, it seems to be not recognizing the wild card here. Anyway, we can check that later. So what we'll do is simply we can give multiple values as well. So in batch mode, you can have multiple values over here, or you can even have one single file with containing all of the molecules. The reason why I'm using different files is that it can it can actually do this for individually, and each file name will be used as an individual for storing the result. That's the reason I have used individual files. I'm just gonna give 3 for now, not all of them. I will show you how to do that pattern one in just 1 minute, and simply start it. So batch mode will take one molecule at a time, and even if you have like 100 molecules, you just run the one command and keep waiting for results to come. So it will one by one finish each one of the docking, and once it has finished all the dockings of all the molecules, it will write this information into that directory that we had created. So let it finish the last as well. So let's go back to the results. So here you can see that for each one of the molecule that we screened, you have the output file with the energy and everything written the same way. So this batch mode can help you. The asterisk sign, basically the common pattern, it works very well into the Linux environment, or the if you have the Ubuntu installed on your Windows, that will be easy. But for Windows, I need to check what would be the wildcard that we can use in this case to find this pattern directly. Anyway, so this way you can use this batch mode. It is much more easier than writing any script or for loops or any other thing. Now this is about performing the batch docking. I will very quickly check what could be the pattern for the Windows and show you the same with simple way, because if you have 1000 molecules, you cannot afford to write each one of their name. You need to have something which is simply common. Now let's go for the next type of docking, that is the flexible docking that you can easily perform with the help of these. The PDBQT extension is missing in the input names. Where are they missing? I think, yeah, they are PDBQT files only. How can we add each ligand molecule if the library contains thousands? No, that's what, you don't have to do that. You can actually give a simple pattern, and I will show you how to do that in Windows. I'm not sure, but the asterisk or the wildcard works easily with the Linux based system. So I'll show you for Windows as well. How does that work? Okay, just give me 1 minute. I'll do that. So this is a simple way. Now, even if when we ran this command for 5 molecules, it doesn't matter even if you have like 100 molecules or 1000 molecules in a file, you have a single command to optimize the molecule and convert all of them into PDBQT files. So that's what you can use. What we can do on Windows is try using a single PDBQT file if it actually does molecule.pdbqt, and then we will use it into this particular command in case it can take all of them at once. No, it actually cannot take a multi-model file, so that's okay. We will look at that later, but we can do that with wildcards. Now the next type of docking it easily provides you is a simple flexible docking, which is also very important for your different types of docking protocols. So first of all, what is a flexible docking? In this, you can allow a certain part of your protein to be flexible, or you can create certain residues which can have a different rotatable bonds or rotational degrees of freedom, which can then be used during the docking. This allows you to explore if your molecule is making any changes to the protein's binding site, or any residues are flipping or rotating in a certain direction to enhance these interactions or not. This can give you a better result. Now that we are now going forward to a different other types of docking, we need to introduce some other tools which will be doing. So now we are going to use this tool that we downloaded, we showed you earlier, that is, yeah, AutoDock Vina's scripts. These scripts can be used in order to perform different flexible docking preparations, water preparations, zinc preparations. So all the scripts are there in this. So either you can download this entire folder and use some of those things. So what I'm going to do is download this entire zip folder into our demo folder and show you what if those are important. It's a very small set of ready-made scripts. I will also explain what are these scripts used for and what can be done. This is a zip file, so I will extract it over here in the same demo folder. Okay. So now, as you can see here in this AutoDock Vina developer folder, you have various folders. One of them is example, and in that you have a folder called as AutoDock scripts. So these scripts are very useful. These scripts contain the prepare flex receptor, that is the flexible receptor. So if you want to prepare the flexible receptor, you can use it. If you are familiar with the flexibility by using AutoDock tools, you can use that as well. But as I'm showing the pre-built commands over here, we will use this. It also has the grid parameter generation scripts, Zinc grid parameter generation scripts, weight, that is basically wetting the ligand or hydrating the ligand. You have this script, adding the zinc pseudo atoms to the protein. So these are very useful for all kinds of docking. What I'll do is I'll simply copy all these scripts and I will put it into our main demo folder, or I will create actually another folder here so we can access all of this. So let's go ahead and create a folder called as flexible docking so we can learn about the flexible docking. So for flexible docking, we will use the same protein, same configuration, just that we will use a different flexible receptors out of that. So I'm going to take the protein PDBQT, ligand PDBQT, and a configuration file. These three files only we need, and I'm going to put them into this folder called as flexible docking. So now we have all the scripts necessary. We only need actually one script, but I anyways have copied all. So let's see how to perform the flexible docking. First of all, how to decide the residues that can be flexible. Well, that often is decided by the residues which are there in the binding site, which could have different degrees of rotational degrees of freedom. So here you can see that the binding site of this ligand contains a lysine, which is on this side, which may be flexible because it's a very flexible molecule. Here we have another lysine. Here we have alanine, but again alanine has a very limited flexibility. We have here asparagine, which could be slightly flexible, and we have here isoleucine. Now we cannot put all of them to be flexible because that will increase the computational load very multifold. So better to decide which one you want to be really be flexible. What I'm going to do in this, I'm going to only use two of them, the lysine 178 and lysine 165 to be flexible, just to see if they have any other conformation better than what it actually is. So in order to get the flexibility incorporated, this in entire setup is again very easy. So first thing you need to do is you need to divide your protein molecule into two parts, the rigid receptor and a flexible receptor. So how to do that? We already have prepared the receptor into PDBQT. Now to prepare the flexible receptor, we need a Python installed. Remember that all the scripts which are available here with the AutoDock, they all are written in a Python version 2.7. So if you want to use, you need to use the Python 2.7 version only. If you don't know how to use it, or you don't know where to get the Python from, it is automatically included into the ADFR which we have installed already. So you should be able to access that. Let me check if my Python is accessible or not. Yes, so directly python.exe from the uh, ADFR should be accessible. If you do not have any other Python version, this is easier. So to run the script, we'll use python.exe. The name of the script, which is prepare flexible. Okay, let me change the folder to flexible, and then we can use prepare flex receptor. Again, it's a very simple script. Only thing it requires to give is a receptor and specify which amino acid you want to keep flexible. So if you have multiple, you have to simply give them in this way, like arginine 8 underscore isoleucine 84 and so on. So you can give as many as you want in this case. If you have multiple chains, then the format is slightly different, but for now we only have single chain. So let's use a simple format. So the way to do that is give the Python name of the script, receptor file name, which is protein.pdbqt, name of the amino acid we want flexible. So we have checked that we have lysine 165 and lysine 178 to be used as flexible. So lysine 165 underscore lysine 178. Okay, make sure that these numbering are very proper according to your structure. They are not wrong, otherwise it will not perform correctly. So that's the only thing you need. The moment you enter the script, okay, there is a memory issue anyways. So I think this may be too much of rotatable bonds, so let's use one at a time. All right. I got it. Okay, it using the actually different Python version in my case because I have many different Python versions installed. So don't worry about it. But what I'm gonna do is I'm gonna specify which Python it should actually use from my computer. But you don't have to worry about it if you only have the AutoDock flexible receptor installed. I'm gonna specify that use the Python version coming with ADFR. That's the only thing it should use. Prepare flex receptor name is protein.pdbqt, and the residues are lysine 165. So once you run this command, oh okay, I think my memory is being used by something, so it's giving the memory error. Anyway, we can correct that later, but this is a very simple way to specify, or maybe we can try using a small molecule asparagine 234 as the Okay, so this has worked. I think the lysine is extremely flexible, so it does take some time. So asparagine we have given as a flexible. Now, if you check the folder, you will have two new files generated. One will be called as what is the name? Protein underscore flex and protein underscore flex rigid. So these are the two things we need in this folder. Okay. I think it's still giving the same error. Anyway, so alanine actually won't have. I need to stop some processing that is using most of my memory. Some simulation is actually going on in the background, so that's probably causing. So this essentially generates two files. Okay, I think I already have done somewhere. We can, yeah. So it generates two files. One will be automatically called as a flex and rigid. So rigid file will contain all the amino acid that has been selected as rigid. That is a normal standard receptor. While the flex will have a file which contains only amino acid which are selected to be flexible. So here you can see that the format of this is very similar to the ligand. Because ligands are by default very flexible, this also will be treated as a ligand in this and then used in the docking. And after this, it's very simply going to do the same thing. Only difference is that now when you will run your Vina, you need to use the receptor for your normal rigid file. Then there is a option called as flex, which you can use to specify the flex file. I'm just giving the name, I'm not going to run it, which will be produced after giving the command. So by default, the receptor is going to be rigid, but if you want to include flexibility, this is what you can do. This flexibility is a side chain flexibility, so only side chains of the amino acid will rotate. The backbone of the protein will not change. That type of flexibility is called as a induced fit docking, where the entire backbone is allowed to move, which is not the part of the discussion today, so we will not talk about it. And rest of the things will remain the same once you give this, the configuration file, the ligand file, everything will be same. So this way you will be able to create the necessary environment and get a different result with this. So once you run this command, it will again take a little bit more time because it's flexible docking and give you the result. If the flexible results are better than the rigid result, that means functionally protein may undergo some conformational changes in a binding site for getting a better affinity of the molecule. So in some cases this could be useful. Now let me go through some of the questions. Uh, any way to show the final result score wise, just like PyRX, otherwise checking the score for each? Well, usually we do write some scripts, basic Python scripts to fetch these results and prepare the tables and all. For that, I think PyRX is the only good way if you want to do it. Otherwise, I'm going to upload this script soon on the GitHub page. I will share with all of you all these new things that I'll be adding up soon. If you have 100 molecules that are drawn separately in SDF, how to combine? Again, it's very easy by using the open babel. You can use that. So what you do is you don't give the name of the input file, just simply say star.sdf as an input, and it will combine all of them into a single file for screening if you want. But if you don't necessarily have to combine them, you can directly use it as it is. What are the differences between flexible docking and simulation? Well, flexible docking is just giving a very limited flexibility to only one, two, or maybe three residues at max. Simulation is a complete dynamic study, so you are allowing the entire system to be having the full flexibility. Another difference is even though we are doing flexible docking, it is still in vacuum just like any other standard docking protocol, while simulation is performed in a proper solvent environment, a correct chemical environment to get the better understanding of the stability of those interactions. How to do with metalloproteins docking other than zinc? Yes, I'll come to that just in a second. Will this be uploaded? Well, yes, it will be available at the same link that you have followed. So it will be there. You can follow that again. What is the difference between flexible and rigid docking? Again, the same. In rigid docking, we do not allow any part of the protein to be flexible. Whatever conformation has been given, it is kept the same way. While in a flexible docking, we can allow certain amino acid to change their orientation. Now, if you want to see the implication of this flexibility, which might be there on your docking result, it is useful for certain proteins. It may not be always important, but I will very quickly show you what effect it could have on your results, how you can improve by using this flexible docking. So let's take an example of this protein here, which is called as alpha murmurin. So if you look at this protein, this buff or a kind of brownish color protein is a unbound protein when it was not bound to the ligand, that is a crystal structure, while the blue color protein is the protein when it is bound to the ligand. You can see that in a crystal structure, upon ligand binding, this particular amino acid, that is tyrosine, it undergoes a certain transformation to remove the steric clashes here and to create a new hydrogen bonding interaction here. So this is what happens naturally inside the chemical reaction. So if there is a possibility of such things happening, if you do the rigid docking with this molecule, it will not give you correct pose or correct result, because what will happen now, this will create a steric clash because it is fixed. But if you allow this residues, all the residues to be flexible, it can explore different conformation where it can have a better pose or better binding affinity. So for the same molecule and the same system, when I ran the rigid docking, it gave me affinity of negative 8.4, while when in multiple runs, when I did the flexible docking, it was slightly improved to negative 8.9. It can be even more improved if it is there the case. So that's what the difference between the rigid and flexible docking is. Now, okay, I think I'll just close some of the programs and I will start doing this quickly again. But now that we have seen how to run this codes or how to run this very specific type of docking, let's come to some more types of docking which we can explore. So the next type of docking that we are going to explore in this case is going to be a metal ion docking, which is another very useful thing which is provided by that. We also have a simultaneous docking that can be done, so we'll talk about that as well. Okay. All right, so let's first talk about the simultaneous docking, which may be very useful for fragment based drug discovery. And then we will talk about the zinc as well as other metal ion dockings that could be possible. I think we won't have time to go through too many of these things, but let's just do one or two more and then we can take a Q&A about all the other things quickly. So now let's do the simultaneous docking. So first of all, what it means, when we want to dock simultaneously two different ligands into the same binding pocket, we use that. So to simulate these fragment based drug discovery, what I will do is I will manually drop the bond between these two uh, atoms over here of this ligand and create two separate fragments, and then I will dock these two separate fragments as two separate ligands. The correct result would mean that we should still have the similar conformations of both of these, and then in order to create a new molecule we may be able to combine them. So I'm going to delete this particular bond at this point. So now we have here two options, two different separate fragments which are there in the binding cavity. I will close the protein part because we don't need it at this point. And I will save each one of this fragment as a separate ligand. So first I will select this fragment, file, save PDB. Let's create here new folder and let's call it let's create a new folder in our demo so that we can store the results separately and look at them later at the analysis part. So this new folder, I'm going to call it as fragment best, or maybe let's just call it as fragment docking, and let's dock this simultaneous ligand into single. So what we'll do is this, the first fragment, we will save it as a PDB file. Let's call it as fragment 1 and save that. Then we will invert the selection to select rest of the fragment, the second fragment. Let's save this as fragment 2.pdb. So if you check the folder now, you will have here two. Oh, okay. Where did I save those? I think I saved in a different folder. Oh, okay. I saved in the main demo folder. So let's get them from there. Okay, so we have here two fragments, fragment 1 and fragment 2, which will put into the fragment based docking. Our protein and the configuration file are still going to be same, so I'm going to use the same files for this as well because we are taking it from the same system, so it is feasible to do that. Now, how to perform this simultaneous docking? So again, it's a very simple. Only thing is that we need to know how to specify those two different ligands. So I will again go to our folder into the demo. Inside that we have a folder called as fragment docking. Here you can see we have access to all the required files. Let's now oh, okay, Vina 2 is in our demo. Okay, so this will be the same receptor specification, exactly same, nothing would change there. Only difference will be in the ligand specification. So now instead of one, we have two different ligands. We need to first prepare or convert them into PDBQT. We can do that quickly. Prepare ligand fragment1.pdb and output should be fragment1.pdbqt. Same way we will do for the second ligand, and then we will run our entire Vina the same way. So to specify multiple ligands within the same ligand flag, you have to write all the ligands. So frag1.pdbqt space, immediately you have to look for the second ligand, that is frag2.pdbqt, and configuration file will be the same again because our protein hasn't changed, so configuration won't change. And that's it. You simply run this, and now okay, I think I forgot to give the output file name. Yeah, so you can also give the output file name where your results could be automatically stored, or you can skip that. I will call it as fragment.pdbqt. Okay, so now it is what it is doing is docking two individual ligands at the same time into the same single binding cavity. This is called as a simultaneous docking of multiple ligands into one binding cavity. Now, what I will do is I will show you this ligand which we had used by breaking down. This was the original structure of the ligand, which was a complete. And now I will open the result of the docking. Obviously, it will not be accurate because we have not increased the exhaustiveness. But let's anyways open the results from the fragment-based docking called as fragment.pdbqt. So you can see that for one of the fragment, the orientation is quite accurate. Only for second fragment, there is a slight change or rotation, which can be corrected by increasing the exhaustiveness. So it is docking the two individual fragments into the same binding site, giving you a way of doing fragment-based docking or fragment-based drug discovery if you want to do that. It has a lot of applications, and it is also another approach, alternate approach to perform a drug discovery, just like structure-based and ligand-based drug discovery. So this way you can do multiple docking. Now let's come to another important type of docking, which is the docking with the metalloproteins. There are many proteins in our body which are metalloproteins. That means they contain the metal ions in their active site or in the structure. So you can, they are actually very important for overall binding of the ligand or strengthening that binding or their activity. Now, the common molecular mechanics force fields that we use for these protocols, they do not have correct parameterization for these metals. The reason is that the metals, along with the other general interactions like hydrogen bond, van der Waals, and so on, metal complexes or metal ions can also create a coordination chemistry. They can create a coordinate bonds with different geometries with the other atoms in the vicinity. So to be able to understand those, the parameterization needs to be done correctly, and that's where we use this metal protocols. Well, the AutoDock protocols are actually optimized for the zinc, but even if you have other metal ions, it is possible to perform it, but you have to manually do some constraint analysis and apply some constraints. To give you a very quick view of what I mean by all that, let me show you a quick diagram of what AutoDock Zn means. So when you have the zinc in your structure, so zinc can make certain type of coordination along depending on what other atoms are present. So for example, if it has a tetrahedral coordination geometry, it has certain spaces in the 3D space, only there it can make the contacts with the ligand. So in order to be able to understand this geometry and use them in your protein, you need to see that where is a possibility of your molecules to be creating these bonds and so on. This you can do with chimera. Again, it has a simple way of visualizing the coordination geometry or coordination complexes of the metals with the ligands as well as the proteins and so on. So for example, let's say if this is the zinc ion and these are the four spaces where it can have connections. Let's say these three are already occupied by the protein residues or protein atoms from amino acids. So we have only one space in the 3D where if the atom is present, the coordination can happen. If the ligand atom is present anywhere else, the coordination cannot happen. So what we need to do is we need to add a pseudo atom at this location so it can be used as a way of attaching to the ligand, and that will help us generate the necessary coordinate geometries. So that's why considering the metal geometries is very complex, but for zinc it can be done easily with the help of this AutoDock Vina with the force field. Now for zinc, we are going to need some extra preparations as well. So I will go over them very quickly in next few minutes, and then we can try directly show you the results and go through those. So what all things we need? The first thing we need is to prepare our ligand normally, just like we prepare. So what I'm going to do is I will take one file here which contains the zinc. So when we downloaded this AutoDock Vina, it has some proteins which contain the zinc. I'll also show you how to actually look for those uh, proteins and some basic data about it. So first of all, let's look at this particular protein and see where is the zinc and how does that look like. Okay, so when you open the protein in Chimera, very quickly you can easily identify that this is where the zinc atom is present. So now this is the zinc, but you're not able to understand what kind of coordination chemistry it has, whether it is binding to ligand or not. Well, in this case, it may not be, but let's just check anyways. To understand the coordination chemistry or geometry of the zinc, you can press Ctrl and double click. It will show for coordination geometry. Here you can see that it can have a trigonal bipyramidal geometry with these atoms, and you have tetrahedral geometry with these atoms. You can also see the pseudo bonds that it can create. So where could be the spaces for other molecules to bind. So this is a simple way, but again, we don't have time to discuss all that. Along with that, we also need a file which is called as AutoDock Zn force field. This force field file, it comes with the details of the zinc as well as many other. Let me check the file. Yeah, so let's have a quick look at this particular file. So this file contains, which comes along with the Autodoc Vina, it contains the coordinate as well as the parameters for different types of metals, not just the zinc, but you have iron, that is Fe, zinc, you have calcium, and other. And this is the atom that we want, that is the Tz, that is a pseudo or tetrahedral pseudo zinc atom. So this is basically atom which does not have any properties, like it has van der Waals radius 0, everything is 0 here, but it specifies a position in a 3D space where the ligand can bind. So this needs to be added to your system before you go ahead and perform this kind of an analysis. So to perform this zinc metalloproteins, we will take an example here and perform this. So you need to have a protein which contains a zinc. You need to have your ligand, which is over here, and you need to have access to the orthodox zinc force field, which needs to be added in order to perform. And then you need a script which is already present in this particular AutoDock Vina folder that can be used in the same example folder which I already showed you. The script is called as prepare GPF for Zn and zinc pseudo. So this zinc pseudo will help us add the pseudo atoms to the protein and create new protein file. So we will use both of this to be able to perform the analysis, just to make it easy. So again, I will go to my Windows terminal where I can perform these analysis and simply run. Second important thing you need to remember is that for zinc metalloprotein adding, you have to use the AutoDock force field. It cannot work directly with AutoDock Vina force field. So you need to prepare the grid parameters just like normal AutoDock and perform it. So as we do not have time to go through everything right now, it's already been long time. So I'm just going to directly show you from the website the workflow and what changes happen. So the first command you will give is the Python and the zinc pseudo atom. The command looks something like this. The script name, obviously it has to start with your Python interpreter, whatever you have installed. Zinc pseudo protein tz.pdbqt. So what this does is that it creates a new protein which contains everything just like a protein, but along with that, where there is a possibility of binding, it also adds an pseudo zinc atom. If there are two possibilities, it will add two zinc pseudo atoms depending on the geometry. The default geometry is tetrahedral. If you want to use something else, you have to do that manually. So here you can see that the charge on this is completely zero. The coordinates are given and the name is TZ, that is tetrahedral zinc. That's how the new protein will be prepared. So it has an extra atom which you can visualize even in visualizer. And that's it. Then you go ahead, prepare your ligand just the way we normally do. The next thing, we need to generate the affinity maps by using the other script and run the Vina with AD4 scoring function. So once you do that, then you will have the result. So I will directly show you the result of the this particular run that already has been, and how does it look like. So now in this particular binding mode, if there is any connection with the zinc which is possible for any atom of the ligand, it will also be shown. So that's how you can perform. Now I know that I don't have time to go through all of this right now, but what you can do in case you find any difficulties, the in the AutoDock Vina, they have this link called Read the Docs, where all of these different protocols have been explained. There may be some difficulties in setting up, but I think you should be able to overcome that once you do this basic docking the way I have shown. So you can follow that as well. For the Python based everything else, I will take a separate session. So only for those who are interested in Python, you can attend that and learn about this advanced docking like Python integration, automated analysis and post processing, generating the energy tables, filtering the compounds, docking with the water as well as macro cycles. For that, you need to have Python installed. But without Python, till the hydrated docking, you should, sorry, zinc docking, you should be able to perform all of these options. Okay. So I think we will stop here for this particular session. It has been just introduction to this particular field. So go over it again. Let me know if you have any queries, questions. I will see that batch script thing meanwhile. But meanwhile, you can post your questions. Another thing is that if any one of you is interested in learning about the complete molecular docking from the very basics to the advanced state with complete AutoDock suite, we have a workshop coming up in the December, which is molecular docking and dynamic simulations. It's a 12 days intensive hands-on workshop. You can also opt for only docking or only dynamics if you want, and it will cover all the things starting from the foundation of the basic scientifically correct preparation of your molecules, titration and protonation in the pH dependent way, and all the necessary things, finding the binding sites, predicting those so much. So all of this can be learned. So any one of you is interested, you can visit our website. I will post it as well in the chat box, and sign up for that. Let me know if you have any questions about it. Yeah. Any other questions you have, just post it in the chat box and we will now discuss. So we will now go to the Q&A session. Meanwhile, I'll try to figure out the batch mode issue that is not detecting this asterisk as our uh, wild card for this. All right. If any one of the questions were not answered earlier, please remind me of that as well. So I'll try to address those also. So thank you so much everyone for listening patiently and asking very interesting questions with this. How to deal with the metal complexes as ligand and proteins having metal ions other than zinc. Okay, if your ligand molecule has a metal ion, AutoDock directly by default is not capable of handling those. It will either remove that or discard it in the process. If protein has ions other than zinc, you can follow the very similar procedure, but what you have to do is it's a very elaborate procedure. Let me give you the link for that as well. One second. You have to manually add the parameter, generate a parameter file by studying the proper geometry and the energetics of that particular atom, and that add the new parameter file to this. So currently their site website is being updated, so they don't have access to that entire protocol right now. I may have saved it earlier somewhere, I'll share it with you. But just like Zinc, okay, let me see. There used to be a very simple link to perform that. So any other thing you should be able to add. I don't remember the link specifically. Yeah, hopefully it opens up. They have actually, anyway, so what you can do is you can refer to this user guide, that is AutoDock user guide. So in their appendix, they have given how exactly the parameter file should look like and what are the different parameters you have. So any other type of uh, atom that you have and you want to kind of uh, prepare for that, you need to generate this force field parameter file by yourself. It's called as DAT file. Where it is. Okay, I'm sharing the link for the user guide where you should be able to get that. So appendix 1. Somewhere in this, just find out that you will get a way of writing the DAT file. So that file has to be exactly written for your molecule, your atom, but that needs to be known to you, that what kind of energy, what kind of van der Waals radius, what kind of effect does it have. And then, but you should be able to find that in any literature. So you can just see if somebody has done that, use the same parameters and generate the parameter file and just include it into the same folder where you have all the data. That will automatically include the parameters from that file and use it in the calculation. Sir, I am interested but can pay in 2 installment. Yes, you can do that. Just drop me a text or email later. I will discuss how to do that. Start should be first for batch mode. Okay. Let's see if that works for the batch mode. That's a good suggestion. That could be a possibility. But dealing with this in a Linux environment is much more easier with the Windows. Even Python setup, it's very difficult in Windows than the Linux. So let me go to the I think batch mode we have here only. So I will go back to the command we were using for batch mode. I think I have closed it. Anyway, Vina receptor. Instead of a single ligand now, we have a batch ligand. So we have to give batch. Our names are molecule. So let me try this way if it works. There has to be an easy way here as well, just like Windows. Molecule. Anyways, I think I'll need to just figure this out, or how does it work on Windows. and then I can get back to you properly regarding how does this work, or I can show you that in a Linux based environment the same command how is going to work. So let's do that. Cd desktop demo, and then let's copy the same command. Only thing we are gonna have, what I'm trying to do is show the same batch how it works in a Linux environment. The command is not gonna change much, but it should be able to follow. Have I given the flex residue? Anyway, I think I'll get back to you on that and show you that. Okay, anyways, so thank you so much, and I hope you have got some idea about what are the new things that you can do with this new version. Obviously it's still under active development, so there are many new things expected in next couple of days, maybe months, so you should expect better from this. uh, there are few questions. Will we get the recording of the lectures when we attend the upcoming workshop? Yes, every day the recordings will be shared with you, so you can follow them up and practice with those. So, is it not possible to use AutoDock or AutoDock Vina for docking metal complexes as ligands? No, by natively it's not, but you can do a very elaborate manual modifications to these codes. There are some scripts that you can use which already exist, and then use your metal complexes even in the ligand. But as I mentioned, okay, in the workshop, in the upcoming workshop, I will definitely try to include that as well, how to prepare, because it's a very elaborate procedure, so we don't have time for that. But what you need to do is all the atoms which do not have any inclusion in the default Autodock parameter file, they can be parameterized individually, and then once you have those parameters, just include that file. So even if your ligand contains a metal, it will automatically detect its energies and its bonding patterns and everything from that parameter file, and will perform the calculation. So all you need to do is correct parameterization and you are all sorted. Okay. All right then, guys. So thank you so much. Let's stop here for the day, and hope to see you again in some other classes. This particular session, I will continue with the complete thing, that is the Python integration as well. So that time also I will again include the batch mode. I just need to figure out how does that work on Windows system, and then we'll get back. It was not that difficult, but let me see anyways. Okay, goodbye everyone, and take care. Stay safe, stay protected. Yes. Zinc, anyways, is a part of the workshop because we always take that. But other metals also, I will include that in the workshop, how to create the parameter file, because anyways, we're gonna talk about other advanced things as well, like induced feed docking, where we can actually perform the backbone corrections as well, and so on. So as a induced feed docking also is very important in some cases. So as I showed you example of this protein alpha membrane, similarly here you have a different protein called biotin. If you look at this, the normal structure of the protein and the structure which is bound to the ligand, it has a complete backbone which is moved towards the ligand to enhance the binding. So these kind of movements, loop movements, are very useful to detect if your protein is flexible. So this can be detected by induced fit or docking. So those also we will discuss. Okay. So do try it out, this new Vina version. If you already are familiar with the old Vina version, see how much better things you get, what changes can be done. Is it possible to do covalent docking with AutoDock Vina 1.2? No, for covalent docking, you have to perform normal AutoDock calculations. Vina is not currently, they have not added this functionality yet. So for that, if you want to use, you have to go for uh, AutoDock website only. Uh, Yep. So on their website, they have the detailed protocol of a covalent docking given. Again, it's a very limited. It's not like a very big thing. You have to actually know which atom pairs to be used for potential docking, and then you have to do that. So on this link, again, I'm sharing the link for the covalent docking here. And in this you can see the entire script files and tutorial as to how to set. It's a very again elaborate protocol where you need to specify the potential atoms and the xy maps like a pseudo atoms which can be connected. So it will use it to form the covalent bond and accordingly generate the conformations as well as calculate the energies including the covalent bond energy. So though limited applicability, it can be extremely useful in many cases. Like for example, aspirin, when it binds to its target, it does make the covalent bond. So it could be easy to study them. How do we decide a perfect pocket for a protein for docking? Well, if your protein already has a co-crystallized ligand bound to it, some strong ligand, that's probably the binding site that you're looking for. If there is no information present about any binding pocket or any active site, usually literature sometimes specifies that these are the amino acids which are important for its activity, so those could be your targets as a binding pocket. Even that information is not available, then you can use many prediction software like AutoSite or AutoLigand or CastP or Pocket Finder. There are many such algorithms which can detect the potential binding sites, or Discovery Studio has a binding site finding tool. So those can be used, and the most preferred pocket you can start with Explorer for the docking. How to convert smiles to multiple PDBQT? Again, is the same command as I showed earlier. That is the open babel. So if you have your input in a smiles file, so the command will be simply changing to obabel hyphen sm input smi or smiles. I think smi is what, and the name of the file, molecules dot smiles. Output should be PDBQT, so it will perform the PDBQT conversion into the same way, and the name of the output file that you want. So that's all. So here, no matter which input file you have, you just have to specify, or you can directly use the file name also without this specification. Just I should be enough. So you can use SDF files, mol files, PDB files, mol2 files, any files you have, you can directly convert to any other format. But always remember, specifically for smiles, perform the correct conversion. I'm going to upload a separate video only for OpenBabel. OpenBabel contains many useful things like, uh, OB conformer, OB tautomer generation, then OB minimize, that is minimization and optimization. So all of these can together be used to generate a really good quality 3D structures of your molecules. You can check for chirality and many other things you can deal with OpenBabel during the conversion, so that you can use for smiles, because smiles are by default 3D, and if they are very simple smiles, do not contain the chirality information or the optical isomers, then it could be a problem. So deal with that by using this variety of OpenBabel commands together to prepare them properly. The Miko command that I mentioned earlier, which comes along with the AutoDock Vina, but you need to install it inside the Python package. So if you want to prepare or protonate your molecules at a certain pH, you can simply specify the pH with the Miko command, and it will automatically protonate and generate the necessary protonation states for that. Okay. All right. Any other queries, any questions? Do let me know. You can comment on the video again or drop us an email with your queries anytime if you want to, if you need to know something about docking, molecular dynamics, or structural bioinformatics in general. I'm also sharing our email ID, which you can reach out to in case you have any queries. I'll try to answer as soon as possible if I will be able to help with that. Okay. So that's where we stop here. Do visit our website. See if you like something. If you want any specific courses to be conducted, let us know. We'll talk about that as well. And hope to see you, at least some of you, in the next sessions, next courses. We also try to keep, we are planning to keep these kind of sessions open for everyone also once in a month at least on different topics. So if you have some specific topics or suggestions you want to know about, just drop us an email with those. We will consider those for the next open sessions. From which site I can download AutoDock Vina and which version, please? So to get the AutoDock Vina, the main website of AutoDock Vina is this vina.scripts.edu. When you go to the main website, which describes everything about it, on the right hand side you will have the latest version of AutoDock Vina is version 1.2. Just click on this link, it will take you to the GitHub page. So here so on the right hand side you will have your button called as releases and the latest release 1.2.3. Just click on the latest release and it will take you to download page. Okay, and depending on which system you're using, simply download that executable and use it in your studies. If you don't rename the file, you can directly use it as an executable. If you rename the file, remember to put .exe, that is executable, as an extension so that it works properly. If you don't rename, it's fine, it can work directly. Okay, got it. How, where to download the version? Just try it always for the latest version, which whatever is available. Keep checking also periodically that whether the new version has come or not. All right, everyone. So I think if there are no more questions, we will say goodbye here. I hope you have got something from this particular session, and we'll use the AutoDock Vina new version. Try it out. If you're not able to set up the issues with some commands or anything, just drop us a text with the screenshot or email. We'll get back to you. Goodbye, everyone. Take care and see you soon.